We're excited to welcome back to the show two great friends, BIOS top voices in George Church and Bob Langer. World-renowned scientists, esteemed professors, and serial biotech entrepreneurs. Thank you both once again for joining us. We always appreciate your support. Today, I'm joined by my colleague, Chris Godbond, to help host things. We're excited to dive in. George, if you wouldn't mind kicking things off for us and sharing a brief introduction for our audience.
Yeah, I'm professor at Harvard and MIT. Technology development, mostly molecular biology, improving reading and writing DNA by about 20 million fold collectively. And nevertheless, there is still, even when you bring down the price of something 20 million fold, you don't expect it to still be profitable, but it is. Also, anything you can read and write with DNA, so that includes viral delivery, cellular, aging reversal, organs, even endangered species that could impact ecosystems.
Terrific. Thanks, George. Always a pleasure. And Bob, if you could share the same yourself, we'd love to hear a brief intro for our audience.
Sure. I'm a chemical engineer, and I've sort of migrated into, over many years, doing largely biomedical research. And the kinds of things that we've been involved in and continue to be involved in, well, some of it goes back a long time ago when I was a postdoc in the 1970s, developing some of the very earliest nano and microparticles that could deliver large molecules. And we continue to do all kinds of things on various drug delivery systems, both in the lab and in companies that I've helped get started. So that's been one big area, and the other big area has been tissue engineering. I mean, trying to grow new tissues and organs from scratch, whether it be for people or on ships to help study, different types of drug development and things like that.
Fantastic. Thank you both once again for joining us. I'll pass it off to Chris now to dive into our first topic here.
Thank you, Chaz. You are arguably the most prolific bio-entrepreneurs of our age and serve as leaders of academic labs that have mentored innumerable founders and professors. You also have track records that include the founding of over 40 startups each, collectively almost 100 companies. Not to mention launching entire fields of biotech, just as you mentioned, Bob, with drug delivery and tissue engineering, and also synthetic biology. So Bob, to kick it off and bring it to you, as someone who's built the largest biomedical engineering lab in the world, what are some of the principles you would recommend to PIs who are building their labs today?
Yeah, well, I guess I'd say a number of things. First, and I think George certainly is a great example of doing that, is to ask really big questions. A lot of times people get scared when they start out and they want to do incremental, what I'll call incremental research, because they feel that's the easier way to get grants. But I just think that what you want to do is ask really big questions that could have a transformative effect in the world. And they may, you may get rejected in your grants some of the time, maybe a lot of the time, but I think it's worth it in the long run. I think a second thing to me has always been hiring great people. Like a lot of people feel like you want to hire somebody that's got an almost a specialist background in the particular area you're working in. And I've repeatedly not done that. I've hired people who I think as postdocs or even graduate students that, are what I call great athletes, but they may not be specialists at all. I also think that you want to, again, aim high when you send your papers out, to send them to great places. And I also, believe in patenting whatever you do. And I don't think it ever, from my experience, I know sometimes people think it holds up papers. I don't think so. I mean, what we do is we, write many drafts of the paper. Let's say we write 14 drafts. So I could use draft number 3 as a basis for a patent. And so that's another thing I guess I'd recommend, because the patents will give you leverage later on, whether you want to get involved in starting companies or you want to get grants from companies. And finally, the last thing, maybe the most important thing, is to treat people well, to really make sure that you treat people. I mean, it's almost like the golden rule, but you really want to treat people the way, if you were a young person, a student or a technician or a postdoc, you'd want to be treated. Those are a few. There's probably a lot more.
No, and that's a great starting point. Thank you. And it sounds like there are a number of different points that intersect, which is important. And I want to build on that last one you mentioned, which is around the people. And this is a question open to you both. As we think about drawing on your extensive expertise, building this sort of translational academic lab, and thinking about building the culture behind it, we'd love your thoughts on the most important elements to consider when you build and scale an entrepreneurial lab culture, even as you build the lab.
Well, I'll start, pick up where Bob left off. You know, I agree with everything he said, and part of being nice to your people is hiring nice people. And it seems like, oh, I should be hiring the smartest people, and we do, but nice is high on the list. And another interesting reflection is on, you know, you may have seen scenes from Apollo that where they say failure is not an option. That's sort of their motto. I think a better metaphor for our kind of work anyway is that you should fail 1,000,000 times a day, but succeed a few of those, right? In other words, the metaphor here is the library. So we make libraries of compounds, libraries of viruses, libraries of cells, sometimes in the millions, billions, and even trillions. Bob does some of this as well. And we expect most of those to fail, but that's the point is we've tried a lot of times, and that's a lot better than trying to design the perfect prototype first time and not do anything until you get that perfect.
Yeah, I really agree with that. I mean, that's probably a better way of saying what I was trying to say about asking big questions. Clearly, when you ask big questions, you're going to fail a lot of times. Also, I think it's important to realize, you know, when somebody says, you know, I've certainly seen the news media and other places talk about failure and say, well, this doesn't work, that doesn't work. I mean, I've had many experiences, for example, people telling me nanoparticles would never work. And obviously they've worked tremendously if you just look at what we did at Moderna and other places are doing with nanoparticles. But part of the point that George just made, which I think is just really important, is that just because if you have like created what I'll call a core technology or a platform technology, you'll probably find a number of times that because of the choices you make or other reasons that it may not work. But that doesn't mean the technology doesn't work. And you just sometimes have to find the right people in the right places and the right company. And so I think the idea that which George was talking about failure, I mean, anybody that does things that are going to make a difference, I mean, they're going to fail all kinds of times, and that's okay. In fact, you know, one of the things, I've had different people like ministers from different countries sometimes visit me and they say, how can we make a more entrepreneurial culture? And what I've sometimes said in return, you know, I said, well, one of the things that's about the United States, if you look at it, is people, you know, failure is okay. There's no shame, you know, maybe you don't want to fail, obviously, but failure is okay. Like you know, whether like I think Steve Jobs' first company didn't work out very well. Lots of companies didn't work out very well. One of my favorite stories, because I like chocolate so much, is Milton Hershey. He had a dream of starting a candy store, and he must have gone bankrupt the first 8 times, but not the last time. You know, he became a multi-billionaire, and that was a long, long time ago. But in a lot of cultures, failure, people are ashamed of failure. And I think that what George just said is just so important. I mean, you know, that is part of what enables so many of these companies that we've had to do well, that the leaders feel that that's okay. They'll take that risk.
No, I think that's really well said. And I think you bring up one of the most important points of biological research in general, which is, as you think about these big picture questions, you need to be resilient in actually carrying through and developing solutions. And I guess taking this in a slightly different direction, but drilling down on that point, I'd love both of your thoughts on how do we think about balancing that resilience with a recognition that we might need to take a step back from answering that targeted question and instead develop the incremental steps that will enable us to get there. Because sometimes biology, we are asking questions where we can't just jump from A to Z. And sometimes we need to go A to M to Z, maybe. Does that make sense?
Yeah, it does. I think we're getting increasingly better at taking big steps, or at taking a lot of little steps so fast that you barely notice the little steps. But you shouldn't, just like you shouldn't be scared of the big steps, you shouldn't be scared of the little ones either. You know, this idea of the library where you have a million shots on goal. Some of those will be small steps, but it encourages you to think about lots of very bizarre things that can come out of a library, even when it's designed with machine learning and so forth. They can be quite radical. In fact, we just published a paper where we showed that we could, this is in the stepwise mode, that if you're changing 28 a region of a protein or a viral capsid of 28 amino acids, you can now change all 28 at once. Without endangering the viability, while it used to be change 2 or 3 at once and you'd have 99% dead or more. So I think that's kind of the theme of Dino Therapeutics, which is one of the 4 machine learning startups we started, was that Dino is a rock climbing move where you let go of all 4 of your limbs at once to move, and so. But by all means, also do the incremental.
Yeah, I agree with that. I mean, I think, and I think you have different places that you can do both. I mean, there's academic research and there's also research at companies. And, in academic research, I mean, some of these things take, very, very long periods of time. I mean, but I think that's okay. So I think you can win either way. I think what George said is exactly right.
To build on that slightly, George, in 2018, your lab at Harvard set a record by spinning out 16 biotech companies in just that year. What are some of the most important factors to consider when you're keeping in mind the readiness of a spin-out?
Well, I mean, first of all, you need to take that number with a grain of salt. The goal was not to set a record. It was, just some were overdue and some were premature for the next year. It just happened to cluster together, and many of them needed to pivot again soon thereafter. But the way is there are a number of ways. I mean, one is somebody can come, one or more people can come up and tell you you're ready. You might start getting lots of requests to an academic lab that seem like they would be a burden if you satisfied all these requests. Of suddenly you have a lot of friends, and so you say, well, let's start up a company that can scale this up in a way that would not be distracting in an academic lab. That's one way you find out. The other way is most of those companies were actually started by postdoctoral fellows, different from the way I started, which was mostly responding to VC inquiries. The postdoctoral fellows, they feel that they're ready. They've seen lots of previous examples in the lab, which is one of these positive feedback loops where if you have some experience in the lab, that plays over. And essentially all of the previous postdocs that started companies are very nurturing for the next generation, and they tell them when they're ready and not. And I tell them when they're ready. But some of it is intuition, that kind of institutional intuition and individual. It's kind of like buy low, sell high. It's not an algorithm. It is it's a skill set that you build up over time.
Makes a lot of sense. And Bob, is there something you might want to add in your experience as well?
Well, I just think, I mean, again, a lot of the companies I've been involved with, are also started by students at least play a very key role in it. And I think that's been great because they're super highly motivated. They've worked on something, for 4 or 5 years, and they really want to make what they did as a thesis or postdoctoral project into a company so they can see it make a difference in the world. So I think that's really important. I mean, to have those kinds of people. Sometimes you want to mix them up with people who have had a lot of experience, but I think there's no substitute for having, the people that worked in the lab, do that. I should add, and I imagine George feels the same way. I'm sure he does. Like as a professor, you feel obligation to your students. You want them to have great lives and you want them to follow their dreams. some of them want to be professors, and some of them want to start companies or be in companies. And I think, you know, as professors and as their mentors, you want to do whatever you can to, you know, to facilitate that happening.
Speaking of some of that facilitation, how do you think about then working with other university elements of the technology transfer office around patenting and around licensing the technology out? And maybe we can just start with the technology transfer office, but how do you consider the people to whom you're entrusting the translation of the science? Do you really like to be hands-on? Is it better to keep the postdoctoral fellows involved, to bring in that serial entrepreneur occasionally? Well, I realize it's probably unique to each circumstance, but would love your thoughts. And maybe Bob, you can start off this time.
I think a typical model for me is, I mean, you're right, every story is different, but I suppose what I've usually done, and some of it gets a little, I don't want to say insidious, but I have, like, you know, we do a lot with venture groups. In particular, we do a lot with Polaris and a lot with Flagship. And part of what we've done, but I have students who got their PhDs with me in both those places that are now like senior partners in those places, or even running the places. And so, you know, and they also are pretty close to a lot of the people in our lab. So a typical model might be, either I might reach out to one of them and say, are you interested, and introduce them to the students or postdocs or other professors, and say, you know, we're interested in trying to start a company. But MIT doesn't want me to get involved in the technology licensing. You know, they'd feel that was a conflict of interest. So I never do that. I might make the introduction, but basically I would let the people at the venture firm or the students, if they've left, you know, work with the technology transfer office and do the licensing. And the way, and I guess the way I've always felt about my role in the company is I don't ever, you know, I've kind of have I sort of take the approach that I'm not a control freak. I want to see these things get happen. I want the people to grow. I want the companies to grow. So I like being an advisor, but, you know, I've always told people when I give advice that doesn't mean you have to take it. But even though I think my advice is usually decent, but I feel so. My role usually is being on a board of directors, scientific advisory board, or something else. But other people, you know, whether it's the CEOs who are more business people, and sometimes they've been my students too. You know, they really do, you know, build the company, you know, and I just try to give advice, but I try to make sure that things happen in a way that, you know, moves forward. And I do help in raising money and with patents and things like that.
And I'm sure they greatly value your support. And George?
Yeah, you were asking about the tech transfer office in part of this question. And, you know, Harvard has evolved an exceptional tech transfer office. I'm very grateful to them. And to, you know, they deal with things ranging from marketing to vetting, you know, potential investors I haven't worked with before. I can make introductions to those that I have worked with before, but it's really, and also partners, and making sure that they manage the potential conflicts between. We have a very diverse portfolio with many things that are licensed multiple times, and so to make sure that they don't, you know, they stay friendly with each other, all the companies. Anyway, the technology transfer office took a big step forward when Isaac Kohlberg came. We have a terrific hands-on relationship with Grant Zimmerman. It's just wonderful staff, and they help the students and postdocs transition into leadership roles in the companies. We have something kind of like incubation at the Wyss Institute, that's where they have projects that can later turn into startups, and that allows them to de-risk and to get, you know, high value without dilution. So all of these things allow me to do my main job, which is science advising. So I do much less administration than you might imagine, because I have all these terrific resources at Harvard and the Wyss Institute.
And I'm sure that really maximizes the productivity of your time, which is phenomenal. And as we think about this, and as we recognize that, as you've both described, company building is often an ad hoc process. Are there any common resources or traditions, maybe other elements that you incorporate into your lab to facilitate entrepreneurship and still and enable a more repeatable process?
Do you want to go first, George?
I'll start. I mentioned the culture, the resources of the Office of Technology Licensing. We also have press office, which is extremely helpful for making sure that the Harvard and Wyss names are used only used appropriately. But more importantly, in terms of mentoring, we have a kind of a backlog of previous entrepreneurs from the lab that are very friendly. They act like big brothers and sisters, and that love their younger siblings, even some they haven't even met yet. And they nurture them along, and they tell them where the mines are and the minefields and where the good things are. So that's another amazing resource, and it depends on having this goodwill that Bob mentioned earlier, if you have that in the past. So much so that the resources that kind of spontaneously developed without, I can hardly say I was trained in this, but it kind of spontaneously developed. They've tried to formalize. So some of the people in my lab, like Sufyan and Oliver, have started Nucleate, which is a system that is helping a nonprofit that's helping countrywide, maybe worldwide, young people start up. So in a way, they're kind of franchising, exporting some of the secret sauce, not so secret sauce.
Yeah, no, I agree with that. I think another way I look at what George said, and I think we have one of the advantages of seeing all these companies start for both of us is the alumni, so another side of that is not only do they help the younger people, but they're also role models for them. And I think when you have role models where you've seen people do it, then other people keep thinking, well, gee, maybe I could do it too. And I think that really has been very helpful. Also, one time, I remember for a while, we had these seminars where I'd have people who were alumni come in. Some were in venture funds, some were in foundations, some were in, small colleges, some in large colleges, just give talks about what their careers were like. And they ended up being very helpful to a lot of the, the younger people in the lab in terms of shaping what they wanted to do. I also think that there's certain things that just happen culturally, and that people, because again, they see the people before them having done it. So a lot of people, I've had a lot of people come into my office and say, gee, can we write a paper and send it to Science or Nature? Can we write a patent? I mean, and they're, or can we start a company? I've had all that happen. And I think it happens because the people before them, they saw that happen, and they had good experience, and those people before them had good experiences. So they would like to see the same thing.
And drilling down into this further, because I think we're all in agreement that mentorship, having these role models and fostering the next generation is really critical. Over the years, you've served as mentors and role models yourselves to hundreds of students and researchers, guiding them through and their own career development. And so, George, knowing that there's not just one archetype, what, in your opinion, are some of the characteristics that make a strong founder?
I think that in founding, you need to have, it helps to have some experience. It helps, and in my case, I was lucky enough to, when I just as a starting student, back in the day when there really weren't any biotech companies, it was actually poo-pooed. It was not dignified. You were supposed to be a pure scientist that did pure research. Chemistry and physics were already fairly mature in terms of startups, but biology wasn't. I was lucky enough that one of my ideas made it into biorad, which was an existing, I wouldn't say a biotech company, it was a, a equipment and supplies company. But I worked with them for a while. Then I was one of the first employees at Biogen, which was arguably one of the first 2 or 3 biotech companies. And that experience as a graduate student helped, by hard knocks, you eventually build it up so that by the time you really need it, you have some experience. And I think the same thing's happening with my students, is that they're in an environment, it's no longer undignified to start a biotech company, but they are getting the experience in what the next step is in the evolution of biotechnology, which might be, it might be machine learning, it might be making organs in pigs, it might, but the point is they're getting exposed to an environment where translation is generally the option. In other words, if you want to have an impact, if you want your paper to make it out of the ivory tower, you're going to accompany it, and they're attracted. They come from far away to be in that environment.
And Bob, taking it in a slightly different direction, are there personal characteristics that really enable a PI's success, which is so often represented through his students?
Yeah, well, again, when you say PI, I mean, there could be academic PIs or industrial PIs, but I think some of the characteristics go back to some of the things that both George and I said before. I mean, people, first you want to think about really big things, about really big questions that can make a giant difference. And secondly, you probably, if you do that, and you may run into some failures, you may run into criticism and so forth. I think it's the people that I see that are successful, are willing to take those risks. They're also willing to really walk through walls to make their dreams happen. And I think having that kind of attitude is really key. They really, you can just see they won't be stopped. They're going to just keep going and going for it. And I think some of the things that both George and I said early on about treating people well and also picking great people, that nice people, I think that's also very, very important. Yeah, and you don't want to feel threatened. I mean, if you can hire people who are better than you, that's great. And it's also important to realize that that's going to be a team. When you do things, certainly in a company. I mean, it's, geez, you have business people, regulatory people, clinical people, manufacturing people, scientists. So you really, you have to have a really good team, and everybody has skills in different areas.
Oh, that's a really important point, especially around the considerations of who you bring into the team. Given how important it is nowadays, especially to be well-rounded, and how much of that comes from your academic training, how do you think about advising trainees in their career development? George, do you want to start?
Well, I think it helps, and I think we tend to recruit people that are already multidisciplinary. It's hard to build a multidisciplinary team from hyperdisciplinarians. You can have a few, but it helps to have people that are already, know 2 languages, and even if they're not shared with other people, the fact that they've mastered 2 means they'll bridge the gap. I think it's good to have. It's kind of setting examples. It's not just role models. It's showing that that your peers, the PI's peers, are interesting people too. It's who you hang out with. So I hang out with Bob Langer. That raises my status.
Mine too.
And when we started Editas, we got together the best and the brightest. David Liu and Feng Zhang and Keith Young and Jennifer Doudna, and then I went on to do a bunch of other things with Jennifer. And it looks, it creates a sense of community, a sense of sharing, and transparency very often is an important component of this. And you have to set an example. You can't just say, do as I say, not as I do. So,
Bob, did you want to add?
No, I think that that's good. I think part of it's also realizing that everybody's an individual, and and again, there's no one size fits all. My big thing when I look at students is to follow their dreams, whatever those dreams are, and then you try to give them advice if they want it, to about, if they're having trouble getting an academic job, or, there's certain things you can do if they're having trouble getting grants. I mean, there's certain things you can do. And if they want to start a company, I mean, there's certain, so again, you can give examples of what you've done or introduce them to people who've done some of those things. And I think that helps. But to me, the number one thing I feel as a mentor is to try to be there to help people live their dreams, whatever those dreams are, as long as they're, not going to hurt people.
Oh, a good message for sure. And passing it back over to Chaz, we'd love to dive into your thoughts on a few rapid fire questions around trends and predictions in the field of biotech.
Thanks, Chris. And Bob and George, a few fun questions for you as we wrap up the back half of this episode here. We're going to go through a quick rapid fire, if you can do for us, overrated, underrated, and why. Quick sentence at the end, why you think what. So given all the topics this episode we've covered, would love to hear what you think academic entrepreneurship holds, overrated, underrated, and why.
I think it's hard to say that it's underrated, but it is. That is to say that, I've worked constantly in both the corporate and the academic, and there's just certain things that the corporate can't do and vice versa. In particular, that it's very hard for the corporate environment to take gigantic leaps. And in the academic, your job is to spend money, not to make money. And so you can take absolutely ridiculous leaps, almost artistic leaps. And I think that's a slightly underrated aspect.
Yeah, I agree with that. I think it's coming up. Years ago, it was a lot more underrated. And I think as time's progressed, it's gotten closer and closer to doing better and better in people's eyes. But certainly, as George pointed out, in the 80s, when we start things, a lot of people would criticize it. Today, they criticize it a lot less.
Taking that and expanding on it a bit, there have been a number of really phenomenal innovation institutes established. And George, you mentioned the Wyss earlier. There's also the IGI, the Institute for Protein Design, and so many more. Overrated, underrated, and why?
I'd say they're properly rated. They are very important. I think to some extent, Bob and I had to do it without them for a while, but then once they arrived, they were extremely valuable. They institutionalized, they produced some corporate memory without becoming ossified. But I think it's important that the leadership have relatively low ego, and I think that's true for all three of those that you mentioned explicitly, but still vision and kind of collects innovative thoughts from a small team of PIs and listens to them. So I think that's the key component of those three: the Wyss, the IGI, and the IPD, which I know quite a bit about all three of them.
Yeah, to me, the answer depends on which ones. I mean, in other words, I think some might be overrated and some might be underrated, and so much depends on their culture, their leadership. I mean, again, I'm talking broadly across the United States and the world, but I don't know that you can just generalize on the institutes any more than on the universities.
In our next question, underrated, overrated for tech transfer. How do you feel?
I'll go. I'd say it's underrated. I think it also, I think, is coming up. But gee, I remember the New York Times wrote some article on me. It must have been around 10 years ago, and it talked about tech transfer. And it's funny, a number of people at medical schools wrote me and they said, the dean of our medical school saw that article and they sent it to every one of the professors in the school. And they said, why aren't we doing that? And so I would say even today it's underrated. I think, George mentioned, that Harvard has a very good tech transfer program. I'd say MIT has a very good one. Stanford clearly has had a very good history of it, but a lot of colleges don't. I mean, and they also don't think about it the right way. A lot of times, like the way the colleges think about it is, am I bringing money into the school, because of licensing fees and so forth. And that to me is absolutely the wrong way. The way MIT, Leda Nelson and Lauren Foster and others have talked about it is, what's going to be the impact? Will if you do have, in other words, rather than get, the best deal that's going to bring short-term money in, what's going to make the greatest impact on the world? And because of their attitude, that's in large part why Kendall Square is what it is today. And I think that pays rewards in spades.
And George?
I think in the spirit of rapid fire, I agree with everything he said, plus a few things I said earlier about, I think Harvard has a great tech transfer.
Oh, well said, George. And keeping the conversation in rapid fire, what do we think about computationally paired biology?
George is an expert in that. I'll let him answer.
Yeah, thanks. Yeah, I've been working with computers since I was 10 years old, which isn't so uncommon today, but it was back then. And it's a key component. It's not, I mean, I would say our lab is maybe 60%, hardware and wetware, but there's a big component that's software engineering. And in particular, recently, the machine learning has proved very important, especially combined with the mega libraries. I mentioned libraries earlier as a metaphor, but it's a real thing where the libraries act kind of like a wet computing device, and you can use the machine learning to do cycles of design and testing. And in particular, we've used this for protein design, which has been like a 50-year stretch goal, holy grail. And now it works, and it doesn't even require three-dimensional structure, although it can help. That can help. We can take, I mentioned we could take big steps, 28 out of 28 amino acids, but also it resulted in 4 startups, Manifold Bio, which is protein testing, where you can do thousands, potentially hundreds of thousands of proteins tested in an expensive animal preclinical trial at once. Nabla on antibodies, Dino on viral capsid delivery, and Patch on cis-regulatory elements, RNA, DNA. The point is that this machine learning is really becoming a big component, as is data storage and management of large projects. Increasingly the case with next-gen sequencing and sequence-based imaging, or spatial biology, as it's sometimes called. These things are now involving petabytes and more. So I think this is, I don't know if it's underrated, but it definitely needs a lot more attention. And starting a company without or a lab for that matter, without some component dedicated to computational biology would be naive.
I think that's a good answer. In the interest of time, I'll also just say I agree with that, which I do.
And for platform companies, the playbook's still largely being written, but how do we feel?
I'm a believer in them in general. I think it gives you such, assuming a lot depends how you define platform, but if you have a platform that you can, as John Maraganore once said to me about Alnylam, and I'd say the same thing about Moderna and some other companies, is that it's like a plug and play. So you can use similar manufacturing procedures to, over and over again to, for different molecules. So I think, I mean, I don't want to say that's the only thing you can do, but there's plenty of good ways to create companies. But I think platform companies are, in general, a good idea, depending on the platform.
Agreed.
And George, I feel like I already know your answer here, but how do you feel about SynBio?
I would say the question is there any other way to manufacture? Sometimes people ask me, what can you make with SynBiology? I ask, what can you not make? You know, I think we're going to be manufacturing things like new materials, computers, recording devices, all kinds of things that you normally don't think of. Inorganic materials, SynBio's getting better and better at that. And even natural biology does a pretty good job of it. So yeah, so that's my highly biased answer.
Hey, George, how would you define SynBio? That's always a question that how do you think?
Yeah. So, I mean, different people have different, I would say that it's like synthetic chemistry, except it includes synthetic chemistry in a certain sense as a subset, but it mostly is a aqueous chemistry and biochemistry, and then anything you make from that, which is cells, organs, ecosystems. It is often described as applying good engineering principles to biology. It makes it distinct from the, you know, early capture of the word genetic engineering was at a time where it really wasn't engineering. It didn't really deserve that term. It didn't have standardized parts and interoperable systems and good modeling for how things work. But now it deserves that term, and I think it's intentionally very broad. It can include evolutionary components. It can include the libraries that talked about, computational, computer-aided design, CAD, CAM, all that stuff. All the best of engineering combined with the best of biology and medicine.
Sounds good.
George, we had one of our guests joke on a previous roundtable here that SynBio needs a rebrand like what we went through with Web3. Curious if you have any branding aspects of where SynBio is taking and how you view the future there.
Yeah, I mean, I realize the importance of branding. I'm not sure this needs a rebranding. I think that what happens is what helps more than that are additional killer apps. Every time you get some new category where they say, I didn't even think that was possible to do with SynBio, that is as good as any rebrand can be. Right?
Great. And as we think about bioinnovation and its pace only continuing to accelerate, amazing opportunities coming ahead. We'd love to ask you both for a few predictions. What do you view as the scientific trends you're most excited by today?
Well, I would say of the various things I worked on, gene therapy was the one that worried me the most because I like equitable distribution, cost effective, you know, and that had happened with reading and writing DNA, but I was worried that it was not going to happen with pharmaceuticals and preventative medicine. But then COVID hit, and COVID was a horrible thing, but it had a lot of unintended positive effects. And one of them was, to my mind, a refreshed new way of looking at gene therapy, where instead of dealing with ultra rare diseases where you have $2.8 million a dose, you can have very common things like aging and pandemics. And the top 5 pandemic vaccines were all formulated in a gene therapy way of formulation. That is to say, 3 of them were adenoviral capsid delivery of double-stranded DNA, and 2 of them were lipid nanoparticle with RNA. And that at as low as $2 a dose cost of manufacturing. So I think that that completely changes the discussion about gene therapy, looking for now common diseases like aging reversal related things. So I'm very high on gene therapy and aging reversal and the intersection of those two, because there's such a gigantic market. 90% of us are going to die of age-related diseases. Aging affects almost every disease, including infectious diseases and accidental harm and so on.
So I'd say to that, I mean, I'd give you a couple of answers. I would say, to me, various forms of genetic therapy, which includes CRISPR, DNA, or gene editing approaches, DNA, certainly messenger RNA and siRNA and things like that. The other big area that I think about are various types of cellular therapies, in particular, I'm excited, as I mentioned, about regenerative medicine, tissue engineering. But the other way I think about that is that what excites me is, and George is certainly a great example of this, is doing really basic cutting edge research, curiosity driven research. And the things that will come out of that and that have come out of it often don't have a name. There's no better example, I think, than CRISPR. If you ask people that question, 10 years ago, other than if you ask and you ask them what CRISPR was, I think other than George and Jennifer and maybe a few other people, nobody would even have known the answer, because a lot of the key papers really weren't even out yet. So I think it's really important to keep your eye on what hasn't got a hot name right now.
With that, we have some obviously great things awaiting us here scientifically, but a few challenges we have to wade through to get there. What do you view as the biggest challenges that we still must overcome, George?
So we have, some people will say it's regulatory. I think the regulatory agencies are wonderful. The fact that they embraced, the FDA embraced such radically new technologies and got them through in one or 2 years for COVID is testimony to how they really like the new things. The new things are likely to help them do their job, which is providing new healthcare. So the challenges, I think, are technical. The technical challenges are we're on an exponential, so it's hard to, it's like kind of a first world problem of keeping up with the exponentials, 2 to 10-fold sometimes per year. But I think definitely one of them is the computation is not keeping up. So if biology is going forward at 5-fold per year and computation is going up 1.5 to 2-fold per year, we got a problem. And we have a crisis where it's very easy to collect more data than you can compute on. So that's one challenge. Another one is encouraging ideas before they get to the seed round. We've kind of got a pretty good pipeline after that, but there's a lot of things that it's hard to get government support. It's too early for investors. Philanthropists haven't heard any buzzwords on this topic, as to Bob's earlier point. They don't have a name yet, and I think that's a new valley of death, which is much earlier than we usually worry about.
I think if we're talking about companies, it is a lot of the valley of death things. If we're talking about academics, I think particularly for young scientists at a lot of universities, getting early funding for important ideas is still a big issue. It depends on the exact person you're talking about.
And any scientific challenges you feel like in the field that we need to overcome, either preclinical models, better biomanufacturing, what have you? Curious your thoughts.
Well, I would certainly think, clinical trials, I mean, George is right, certainly in the COVID situation, that moved very quickly. But clinical trials take a long time. And I think there's a lot of things that hopefully someday could be done to speed them up. Whether there's better models, whether they be, to accelerate some of this, like, again, if you could do some of the high throughput stuff that George said, because you had accurate, humans on a chip or organs on a chip. I mean, if you could categorize people differently in clinical trials, maybe by using, proteomic or genomic designs, and but I think that certainly the big bottleneck for companies in general is how slow it takes, and then of course if you're looking at certain diseases, it's having models that are useful. Cancer's been difficult in part because the animal models have been so bad.
And to build on that, Bob, what do you believe is the next scientific breakthrough coming our way? If you had a crystal ball here, where do you think the next big thing's coming from?
Well, that's what I said before. It's what hasn't been discovered yet. I mean, I really think we should keep an open mind. And the second we say it's going to be X, I think we put ourselves, and that we devote our energy to doing that, I think that puts you in a trap. I think you want to do curiosity-driven research that can that you feel will have a big impact. But I don't think it has to be in any particular category. It may come from left field or wherever, and that's good.
And George?
Yeah. I to amplify on that, I think one example of where we prematurely anointed a breakthrough field was Nixon's war on cancer, and a few other wars on cancer since then. I think that every now and then anointing does help, though. So the Genome Project, I think, accelerated something that probably wouldn't have happened otherwise, and that exponential may have been inevitable, but I definitely think that the project both helped and hurt. It tended to ossify whatever technology we had at the beginning, but at the end, it blossomed into next-gen sequencing and nanopore sequencing, and now spatial subcellular sequencing. So the brain initiative, I think, has been helpful. And so initiatives are okay, but predicting scientific breakthroughs is much harder than accelerating engineering breakthroughs, which are those three examples.
Our last few questions here. One we'd love to ask you both as mentors and leading educators. Is there any changes, given the modern environment and everything we're talking about in academic entrepreneurship, that you would make to scientific education today?
Well, one trend that worries me a little bit is a discouraging of high school students getting into real research. I know my lab used to have many more than they have today. There's just all kinds of subtle pressure away from that. I know that I was greatly helped by being able, had essentially my own lab available to me in my high school. I could work on nights and weekends. And I was at boarding school. And I think that would be very helpful because there's learning it from books and tests is very dry compared to actual hands-on experience. So I think that's one thing that, now iGEM is a trend in the opposite direction. It really is giving high school and young college students hands-on experience with synthetic biology. And I think we need more examples. That is also another trend that it embraces, that I embrace, is some of those called human practices or bioethics. It's in order to win the prize in the iGEM annual competition, you need to have a big component of your project that talks about safety and ethical and educational communication components. It's not just about being a gearhead, producing the shiniest gear.
I think all that's correct. I'd add to it. I think really making science teaching better, emphasizing it more, particularly even in the grammar schools, and trying to make it, trying to develop curriculums at that stage in grammar school and high school that are both fun and relevant.
Thank you both for joining us. We'd love if you have any closing thoughts for our audience. Any last words here, folks?
For me, it's always a pleasure. Now I've gotten to see George twice today on Zoom, but it's always a pleasure to talk to him and to see him, and also it's a pleasure to be a part of your show. You guys do a great job, so thank you for having me.
Yeah,
thank you, Bob.
Ditto, same thing. It seems like video is the new in person.
That's
So,
right. That's
nice
right.
being together with y'all. Yeah.
Save here.
Thank
Great.
you both. We've touched on a lot of fun topics today. How can our audience learn more about your work?
We have, well, I mean, we have a website. That's probably the easiest, is just to Google me, and I'm sure Googling George will work very well.
We're not hard to find. That's for sure.
Exactly.
Awesome. Thank you both again for your time. Really grateful to have you back on the show and hope to host you again soon. Thanks, everyone.
Thank you. Bye-bye.
Take care.
Bye.